Okay, are there any questions? It looks like not too many people are going to come on today, but we will begin. It's a minute afterwards. I'm going to be moving pretty rapidly today, but hopefully you've had a lot of this. It's microbial metabolism, and hopefully a lot of it will be review. Let's see. Oops, I just showed you what I was talking about, and I didn't share the screen. All right. Now I've got my screen going. So like I said, we mostly, it's going to be microbial metabolism today. I'm going to try and go rapidly through it. Hopefully a lot of it will be review. If you look on the schedule, microbial metabolism all this week. And also this week on Wednesday, you've got quiz 3. It will be covering chapter 4 and the end of chapter 3, as well as labs 5 and 6, which we did last week. Any questions about what the quiz is going to cover? In the lab today, we'll be discussing lab 7, and I'm going to start and hopefully get started the unknown project. Oh, I'm sorry. Don't have this on. Hopefully this is a little better. If not, just let me know. All right, let's close that down. And let me mention briefly that in the lab, I don't have it shown here, that I got lab 5 graded, and the lecture to chapter, or last chapter, whatever it was. And so you have until Thursday to make corrections on those 2 assignments. I did not get lab 6, I think, graded, and I'll try and work on that tonight. So you'll have more time till Thursday to get that work done. Any question about any of that? Let me stop showing the screen, and then I'll have an easier time taking a look at this, because I'm just going to look at the grade book. And I can't show you that, so yes, I got extra, the worksheet to lecture 4 graded, and then lab 5 graded. I need to work on lab 6. And I have not gone back to finish lab 4. A number of people who put in corrections, and I have not got those graded yet. But I want to work on lab 6 so that you can get that finished. All right, any question about any of that? No questions? Let's continue. Share my screen. And I thought I had, oh, there it is. So we're going to be covering microbial metabolism. The major goals and a rough outline is to know the terms and understand what feedback inhibition is, understand what aerobic respiration is, the steps in aerobic respiration, and how it differs from anaerobic respiration. How is my voice coming across?
We can hear you fine.
Great, thank you. So we're going to be talking about microbial metabolism. Metabolism is all of the chemical reactions occurring within a cell or an organism. And of course, we're talking about microbial metabolism, so that an organism is a cell. You can split metabolism into two parts: catabolism, the decomposition reactions, the release energy, and anabolism, the biosynthesis reaction, and they use energy. So if you have a complex molecule such as starch, it can be broken down into a simpler molecule like glucose. When energy is converted from one form to another, heat is always given off. Heat is just another form of energy. We're not going to talk much about that. But whenever you change energy from one form or another, some of that energy is converted into heat, which is given off. Some of that energy may be used to make ATP. Now, this graph is a little bit, I don't know, oversimplified or downright misleading, and that is, at least in animals, there are only two chemical reactions where a complex molecule is broken down into a simple molecule and ATP is made. This is called catabolism, by the way. Okay? And that is aerobic respiration that we'll talk about, and fermentation, which we'll also talk about. All other catabolic reactions occurring in an animal cell do not generate ATP. So understand that most catabolic reactions do not generate ATP. Even in a green plant, which is much better at generating ATP than animal cells, most catabolic reactions do not generate ATP. Now, anabolism is converting simple molecules like glucose into a complex molecule like starch, and that is an anabolic reaction. It takes in energy, meaning the simple molecules are made into the bigger molecules because energy is added into the system. That energy is usually derived from ATP. When ATP breaks down into ADP and phosphate, energy is given off, which is used by the anabolic reaction to make the more complex molecules. All right, any question about any of that? Metabolic pathways are a sequence of enzymatically catalyzed reactions in a cell where one reaction is hooked to the next reaction, which hooked to the next reaction. For example, molecule A is the starting molecule, so we call it the starting substrate or the reactant. An enzyme catalyzes the reaction, enzyme 1, from molecule A to molecule B. Enzyme 2 can convert molecule B to molecule C. Molecule C can be converted to molecule D by enzyme 3. Molecule D can be converted to molecule E by enzyme 4. And then enzyme 5 can convert molecule E to molecule F. Molecule F is the last molecule made in the enzymatic pathway. So we have a name for it. We call it the end product. All of the other molecules, B, C, D, and E, are called intermediate products. So the starting substrate starts it off, and then we have intermediate products. enzymes convert them from one to another until we get the end product. Metabolic pathways are determined by enzymes. They cannot happen in a cell without enzymes, and enzymes are encoded by genes. Any question about any of that? This slide is showing you a huge amount of metabolism that is going on within a simple bacteria cell. Are you glad that I'm not going to require you to memorize all of this? Okay? No comment. Chemical reactions happen because of the collision theory. The collision theory states that chemical reactions may occur when atoms, ions, or molecules collide with one another. The activation energy is the energy needed to disrupt these electronic configurations and convert the reactants into the product. The reaction rate is the frequency of collisions with enough energy to bring about a reaction. You can increase the reaction rate by increasing the abundance of enzymes, so that'll speed up the reaction, or you can increase the temperature or the pressure. If you increase the temperature or the pressure, you make a collision more likely, and the collision will have more energy and so will more likely form a chemical reaction. Any question about any of that? Here is showing you a chemical reaction mediated by an enzyme. Every reactant has an initial energy, which is the energy within the molecule. And the products also have energy in them. If you start with a larger molecule and make a smaller molecule, you'll have less energy in the products. So this could be something like glucose being converted into CO2 and water. CO2 and water have less energy in them than glucose. The reaction proceeds by going over the activation energy. And the activation energy for converting glucose to water and carbon dioxide is very great without an enzyme. I could convert glucose into CO2 and water by taking a blowtorch and applying it to the glucose. Does anybody know how hot a blowtorch is? It's around 1,000 degrees Fahrenheit. So you have to have a lot of energy added to get enough activation energy to convert the glucose into the products when there's no enzyme. Now, this same reaction happens in your cells all the time. Glucose is converted into CO2 and water. And in your cells, do this with an enzyme, and so there's a less energy hump, and that we call the activation energy with an enzyme. How hot does the cell have to have glucose to convert the glucose into CO2 and water with an enzyme? How hot does the cell have to be? How hot does the cell have to be before the cell can convert glucose into CO2 and water? Well, a cell can't do it at freezing temperature, at least not your cells. The reaction will not proceed at freezing temperature. So what temperature does the cell do that at?
So I'm gonna go out on a limb and say like body temperature, like 98.6 degrees, let's say, something like that.
Yes, that's exactly right. So the point is that the activation energy with an enzyme is considerably less than the activation energy without an enzyme. Because to convert glucose to CO2 and water without an enzyme, I would need the energy of a blowtorch. Okay? I'm just trying to make a point here. Hopefully you understand what point I'm trying to make. Enzymes are biological catalysts. They speed up chemical reactions greatly. And that's really all you need to know. They do speed up a chemical reaction 10 to the 8th to 10 to the 10th fold. I'm not going to ask you about the numbers, just that they speed up a biological reaction greatly. And enzymes are encoded by the genes. Each enzyme is specific for a single chemical reaction, meaning that the enzyme that converts, well, that's a bad example. The enzyme that converts sucrose into converts I'm getting this confused. That converts lactose into glucose and beta-galactose is the specific enzyme lactase, and that enzyme only reacts with one chemical reactant, meaning lactose. Any question about any of that? So each enzyme is specific for one specific substrate. That's because of the binding of the enzyme to the substrate. We'll talk a little bit more about that. And it happens at the reaction site. Enzymes are not used up by a chemical reaction, so once the enzyme is done, it can turn around and grab onto another substrate and convert it into the product. So we say that enzymes are not used up or altered in a chemical reaction. That doesn't mean that enzymes do not change in a chemical reaction, because an enzyme actually forms a substrate complex where the two are physically bound together. This helps orient the substrate and increases the probability of a reaction, and it lowers the activation energy. What it does is when the enzyme forms a complex with the substrate, it's putting stresses on the bonds of the substrate, helping the substrate convert into the product. Enzymes normally end in the name ACE, so most enzymes have ACE as their name, and well, we'll talk about ligase later in the lab, or not this lab, but later in the class. I don't think I've talked about it yet, but we'll talk about that enzyme. You do not need to know these examples. I'm just trying to show you that enzymes normally end in ACE. Not all enzymes end in ACE, but most do. You should also know that enzymes do not work always alone. Some enzymes require a coenzyme or a cofactor. A coenzyme is a biological molecule. It's bigger than a cofactor. Cofactors tend to be, well, actually they tend to be something like iron, zinc, magnesium, or calcium. And the coenzyme gets together with the enzyme to finish the enzyme. I'm just going to word it that way. You do not need to know the names apoenzyme and haloenzyme. We're not going to go into that. You should know that there are some important coenzymes, and you should know the first two, NAD+, or nicotinamide adenine dinucleotide, or FAD, flavine adenine dinucleotide. You should know these two coenzymes because we're going to talk about them today in the when we talk about aerobic respiration. There are other coenzymes, but we're not going to be talking about them very much, and so you don't really need to know them. Okay? Any question about any of that? You can say that an enzyme works like a key into a lock. So the enzyme is the key that specifically binds to the substrate in the active site, and then the two form an enzyme substrate complex that puts stress on the bonds of the substrate, which helps convert the substrate into the product. Once the product is made, it falls off of the enzyme, and then the enzyme is free to bind to more substrate and then proceed with the chemical reaction time after time after time. You don't need to know the number, but the turnover rate is between 1 and 10,000 molecules a second, and it can go up. All right, any question about the lock and key model of an enzyme? And the idea is an enzyme fits specifically with one substrate, like a key fits specifically for one lock. Any question about that? Let me blow this up. Come on. Now, the next picture is showing you a little more complex model of how an enzyme actually works with a substrate. And here's the enzyme, the big blue molecule, and there's the substrate. And there, this is the active site that binds to the substrate. And there the two are forming a complex. What do you notice about the enzyme and the substrate? What do you notice about this picture? There's actually three things you can identify. One is a little tricky, you have to look real sharp. But two are very easy. What do you notice about the substrate and the enzyme?
So again, I'm kind of going to go out on a limb a bit, but I noticed, at least in that second image, the enzyme envelops the substrate. And then earlier, the substrate is paired to exactly one enzyme. So that would be two things. And then let me think about the third thing.
What's the size of the enzyme compared to the substrate?
Oh, the enzyme is quite large.
Yeah, the enzyme is quite large compared to the substrate. I think you got it, and that is the lock and key model doesn't quite work because the enzyme changes its shape when it binds to the substrate. Do you understand that? So this they call the hand in the glove model, and that is the glove molds to fit the shape of the hand. The enzyme changes shape to fit the substrate. Now, if anyone looks very closely right here and right here at the substrate, you'll notice a very slight change. Can somebody besides Wade, identify what's different? All right, I don't want to spend further time on this, and that is the substrate, if you look really closely, slightly changes too. So even the hand in the glove model isn't good enough. The best way to do it is that the hand is a shapeshifter's hand, and it's changing shape to mold the glove, while the glove is molding its shape to fit the hand. And those of you who watch Star Trek Deep Space Nine know what the heck I'm talking about when I say a shapeshifter. That the substrate actually does change its shape slightly when it binds to the enzyme. And then the enzyme changes its shape too. And that's what's putting stress on the bonds of the substrate, helping convert the substrate into the products. Any question about any of that? Excuse me. For some reason, I just had a coughing spell there. There are various factors that can influence enzyme activity. Temperature, pH, salinity, heavy metals, alcohol, radiation, especially UV radiation, can cause protein denaturation. You can also influence the enzyme activity by changing the substrate. Oh, excuse me. For some reason, I'm having trouble today. If you change the substrate concentration, you can change the enzyme activity. Generally, if you increase the substrate concentration, you'll increase the activity in the enzyme. And then you can decrease or destroy the enzyme activity by adding enzyme poisons like cyanide. You can change the enzyme activity by inhibition, and there's two types, competitive inhibition and non-competitive inhibition, which is more commonly called allosteric inhibition. Any question about any of that? We'll also talk briefly about feedback inhibition and why it's important in cells. Sorry about this. Let me drink a little bit of water and see if that helps. So enzymes can be denatured, for example, putting them at the wrong temperature or the wrong pH. Here's an active functional protein, and it becomes denatured, taking on a totally different shape. You'll notice that the interactions between the amino acids right here are no longer happening in a denatured protein. A classic example of denaturing protein is when you take egg whites and then cook them. So the cooked egg whites are actually denatured protein. Why does a protein become denatured at the incorrect temperature or the incorrect pH? Why does a protein become denatured at an incorrect temperature or an incorrect pH?
pH could change the charge of some functional groups and then disrupt the bonding.
Okay, can you get a little closer to your computer? I didn't hear that.
If the pH changes, some of the functional groups could change their charge, and then that could disrupt some of the bond.
Yes, if you change the hydrogen concentration or the pH, you will disrupt the hydrogen bonding, and you may also interrupt the the hydrophobic interactions too, causing the protein to denature. And so what you're doing is you're changing the way the protein is folding on each other and the attraction of the amino acids to each other, denaturing the protein. Generally speaking, enzymes have an optimal temperature where the enzyme works best, and when you go away from that optimal temperature, the enzyme will still function, but it will have less function. And then when you go way away from that optimal temperature, the enzyme will not work. That's true for pH too. Enzymes have an optimal pH. This looks like an enzyme on the skin, where the optimum is around pH 5. And when you move away from that, the enzyme won't work as well, but it will work. And then when you go to extreme pH, the enzyme won't work at all. With substrate concentration, generally, when you increase the substrate, you increase the activity of the enzyme. But that only happens to a point. Once you get the substrate concentration up to about here, the enzymatic activity no longer increases. Why do you get a saturation level when you increase the substrate concentration after this point?
You only have so many enzymes.
You only have so many enzymes, and each enzyme is already now bound to a substrate, so it doesn't matter how many more substrates you add. You can't increase the enzyme activity because all of the enzymes are actively bound to the substrate. So yes, you're correct. Competitive inhibition is where an inhibitor competes with the active site of the enzyme for the substrate, where the inhibitor binds at the active site and then the substrate can no longer bind, and that would be a competitive inhibitor. Some of our drugs are competitive inhibitor, and that's why the drug works. Okay? Any question about competitive inhibitor? The important part is that the inhibitor binds at the active site, the same place that the substrate binds to. A non-competitive inhibitor, which is more often called an allosteric inhibitor, is where the inhibitor binds at a site other than the active site. And then the non-competitive inhibitor inactivates the enzyme. So when this non-competitive inhibitor binds to the enzyme, it changes the folding pattern of the enzyme so the active site can no longer bind to the substrate. Any question about allosteric inhibitors? Feedback inhibition is when one substrate is converted by an enzyme into intermediate A, which is converted by an enzyme to intermediate B, which is converted by a third enzyme into the end product. And when the cell has enough of this end product, the end product then goes and binds to the first enzyme and inactivates the first enzyme. And that way no intermediate A is made, therefore no intermediate B is made, and no further end product will be made. This is called feedback inhibition. It is an allosteric inhibition in that the end product binds at an allosteric site, not the site that the substrate binds to, the active site. Any questions about feedback inhibition? Sorry. Like I said, I'm having a difficult time today. I'm not sure what the problem is. Now I had to blow my nose. Feedback inhibition is fairly commonly seen in cells because if the cell doesn't want any more end product, you'd like to shut down the production of the end product, and a simple way of doing it is have the end product bind to the first enzyme. You bind to the first enzyme because if you make intermediate A, that costs the cell energy. And so you want to shut down the first enzyme. And then, when there's not enough end product in the cell, it won't bind to the enzyme. And if there's an end product bound to the enzyme, every once in a while it'll fall off. And if there isn't enough end product, it won't rebind. And then that activates enzyme 1. So intermediate A will be made, and then intermediate B will be made, and then we'll make more end product. And this will continue until the cell makes enough end product. Once it has enough end product, the end product will, of course, bind to enzyme 1 and shut down the enzymatic pathway. Any question about feedback inhibition? Like I said, it's very commonly seen in cells because it's an efficient way to regulate the production of an end product. All right, let's briefly talk about ribozymes. Ribozymes are non-protein enzymes. To my knowledge, all of them are RNA enzymes. It's an RNA molecule that cuts and splices RNA or some other molecule. Splices means it puts together. Ribozymes function as an enzyme in that they have an active site that binds to a substrate, and they are not used up or modified in a chemical reaction. So this ribozyme can bind to its substrate and then make more product. Any question about any of that? Your textbook author does not mention it, but the RNA in a ribosome, when the ribosome links the amino acids together, we'll talk about that probably on Thursday. When the ribosome links the amino acids together in a growing polypeptide chain, the enzyme there is an RNA enzyme or a ribosome. And that's the only enzyme we'll talk about that is not a protein enzyme. Any question about any of that? All right, if there's no questions, let's move on to the next topic, metabolic diversity among organisms. All organisms, including microbes, require the following: an energy source and a carbon source. Based upon their energy source, organisms can be classified as a phototroph or as a chemotroph. A phototroph is an organism that derives its energy needs from light. A chemotroph is an organism that derives its energy need from a chemical compound. And this can be an organic chemical compound like glucose. That's what we use. We are a chemotroph. But it doesn't have to be an organic chemical compound like glucose. It can also be an inorganic chemical compound like ammonia or sulfate or iron. And we will talk briefly about the inorganic chemical compounds. They're still called chemotrophs, but most of them are a chemosynthetic bacteria. Based upon its carbon source, organisms can be classified as an autotroph if the principal source for carbon is CO2, either in the air or the water. So a green plant uses CO2 from the air, so a green plant is an autotroph. Well, a heterotroph is an organism that obtains its carbon source from an organic carbon source like glucose. And we are a heterotroph. Where we get our carbon is from glucose and the other food molecules that we consume. By considering both the energy and the carbon source, we can define an organism into these four groups. A photoautotroph, and there's two types. Those photoautotrophs that produce oxygen, which we call oxygenic photoautotrophs, and then anoxygenic photoautotrophs. There's also photoheterotrophs, chemoautotrophs, and chemoheterotrophs. Among the chemoheterotrophs, there are two types: those that obtain their energy from an organic chemical source, and those that obtain their energy from an inorganic chemical source. Any question about any of that? If not, let's look at that in a little more detail. The photoautotrophs generally don't confuse students, probably because students are used to photoautotrophs because of green plants. And they have as their energy source light and their carbon source CO2. Besides green plants, we have the cyanobacteria. That is a photosynthetic bacteria that produces oxygen. But you can also have anoxygenic bacteria like the green and the purple sulfur bacteria. And we're not going to talk further about these bacteria. Let's skip these two, because this will be the one that you're most familiar with next. The chemoheterotrophs, they get their energy source from a chemical compound. It may be glucose. That's where we get our energy source from. Glucose or the food we eat. But some bacteria can get their energy needs from an inorganic chemical compound like ammonia or sulfate or a nitrogen compound or iron. These bacteria that get their energy needs from an inorganic energy source, inorganic chemical source, typically are the chemosynthetic bacteria. But you do have to realize that not all of the chemosynthetic bacteria are chemoheterotrophs. Some of the chemosynthetic bacteria are chemoautotrophs. Probably most of them are chemoautotrophs. The chemoheterotrophs get their carbon source from an organic chemical compound such as glucose. And we get our carbon source from glucose. So all animals are chemoheterotrophs, all protozoa, all fungi, many bacteria like E. coli, and then the fermentative bacteria that we use to make yogurt and stuff like that. They're the chemoheterotrophs. Any question about the chemoheterotrophs? Usually, the chemoheterotrophs use the same chemical molecule like glucose to get their energy source and their carbon source. But it doesn't have to be that way. Like some bacteria get their energy needs from iron, an inorganic chemical compound for its energy source, but they get their carbon needs from an organic chemical compound like glucose. Any question about any of that? All right, the chemoautotrophs get their energy source from an chemical, largely an inorganic chemical compound, like hydrogen sulfide, or iron, or a nitrogen compound. And they're chemoautotrophs, so they get their carbon source from CO2. An example of this is the iron oxidizing bacteria, which you may not know it, but I'll bet you everyone has seen these iron oxidizing bacteria. Just take a look at the water container in the toilet, not the bowl, but the toilet tank, and you'll see some yellow sludge on the side of that tank. Or maybe it's a light brown sludge. That is iron oxidizing bacteria. Bacteria that are acting as a chemoautotroph, and they're getting their energy needs from an inorganic chemical compound dissolved in the water, largely iron, and they're getting their carbon source from CO2. Any question about chemoautotrophs? Like I said, you just didn't know it, but you're actually familiar with the chemoautotroph. The photoheterotrophs are the category that gives students the most trouble. The photoheterotrophs get their energy source from light, and they get their carbon source from an organic compound such as glucose. An example of that are the green and purple non-sulfur bacteria. And most of you probably are not familiar with the green and purple non-sulfur bacteria. All right, any question about this? You do need to understand the nutritional classification of all organisms, know the parts in the red. Let me blow this up a little. You can classify all organisms by their energy source into, if they have an energy source that's chemical, they're chemotrophs. If their energy source comes from light, they're phototrophs. You can then split the phototrophs into their carbon source. If they get carbon from CO2 in the air or the water, we call them a photoautotroph. If they get their core carbon source from an organic compound like glucose, we call them a photoheterotroph. Any questions about any of that? And then for the chemotrophs, if they get their carbon source from CO2, we call them a chemoautotroph. And if they get their carbon source from an organic chemical compound, such as glucose, come on mouse, such as glucose, we then call them a chemoheterotroph. And we are chemoheterotrophs. Any question about any of that? Now I need to say that there's a complication, and that is an organism does not act always the same, and it can act and switch its classification system because it's using either a different carbon source or a different energy source. A classic case of this is Euglena. If you put it in pond water, no food in the pond water, well, other than the bacteria growing, and you give it light, it'll act as a photoautotroph, taking CO2 from the air and using light for its energy needs. But if you add glucose to the water and then remove the light, Euglena will no longer act as a photoautotroph, it'll act as a chemoheterotroph, consuming the glucose and using it both for its carbon source and as its energy source. The bacteria can do something similar or even much greater. The mixotrophs are the bacteria that essentially use whatever is available. For example, I mentioned that some of the chemosynthetic bacteria are chemoheterotrophs, other are chemoautotrophs. Well, some of the chemosynthetic bacteria are mixotrophs. They'll use whatever is available. So if you're growing it on a dish, it will use the organic compounds like glucose that are in the dish because it's available, but at the same time, this chemosynthetic bacteria can pull carbon dioxide from the air. And that means it's acting as a chemoautotroph. At the same time, it's acting as a chemoheterotroph. And these we call mixotrophs. They don't always have to pull both carbon sources, CO2 and glucose, at the same time, but they can. Any question about any of that? All right. Nutrient molecules have energy associated with the electrons that form their bonds. In cells, energy is obtained from catabolic reactions and stored in the bonds of ATP. Now remember, for animal cells, there's only 2 catabolic reactions where we can take those catabolic reactions and then make ATP. ATP is an energy carrier of cells because it has a high energy unstable bond. The 3rd phosphate and the 2nd phosphate are high energy bonds. And the energy can be released quickly and easily just by breaking that 3rd or 2nd phosphate bond. Any question about any of that? When we're talking about the production of energy, it's important to realize that they happen by oxidation and reduction reactions. Oxidation is the loss of an electron. So molecule A loses the electron, so molecule A becomes oxidized. Reduction is the gain of electrons. Molecule B gains the electron. So molecule B, we say, is reduced, gaining the electrons. Oxidation-reduction reactions always are coupled. You always have one oxidation, molecule A, and then one reduction, molecule B. Here's an example. Copper plus 1 and iron plus 3 react in an oxidation reaction, and the copper plus 1 is converted to copper 2 plus, and the iron gets an electron, so instead of becoming iron 3 plus, it becomes iron 2 plus. How can you remember which is which? You can remember this mnemonic device. Leo the lion goes grr. Leo, loss of electrons, oxidation. Leo the lion goes grr, grr. Gain electrons, reduction. Any question about any of that? Okay, oxidation-reduction reactions are frequently shortened to just redox reactions. So a redox reaction means oxidation reduction. In biological systems, oxidation usually has a proton, meaning an H plus ion, usually removed with the electrons. Remember that an H plus atom equals an electron and an H plus ion. Therefore, biological oxidations are often called dehydrogenations because we're moving a proton, H plus, in addition to the electron. A classic point in case is in aerobic respiration, where we move an electron from this molecule here to that molecule there, but more than just the electron moves. Once the electron moves, the hydrogen ion also moves, and this molecule becomes oxidized, this molecule becomes reduced. Any question about that? So in biological systems, the proteins are typically also gained with the electrons. Biological redox reactions are used in catabolism to extract energy from nutrient molecules. Highly reduced compounds, meaning a compound with many H plus atoms, are degraded or catabolized to highly oxidized compounds. Glucose, C6H12O6, is oxidized to CO2 and water. And then some of the energy that's released becomes trapped in the molecules of ATP. Generally speaking, highly reduced compounds contain a large amount of potential energy, and glucose has a lot of potential energy. Any question about any of that? If not, let's talk briefly about aerobic respiration. Aerobic respiration is the way that many cells, not all cells, but many cells, undergo the conversion of energy in a cell. The summary reaction for aerobic respiration is C6H12O6, and that you should know is glucose plus 6 oxygen molecules, 38 ADP molecules, and 38 phosphate this means I, inorganic phosphate, is converted into 6 CO2 molecules, 6 water molecules, and 38 ATP molecules. Any question about any of that? So here we're seeing glucose getting together with oxygen, and then in cellular respiration, glucose is converted to CO2 and water. Any questions? Energy production and the generation of ATP. Some energy from some redox reactions is used by the cell to make ATP. ATP is generated by the phosphorylation of ADP. Phosphorylation is just the addition of an inorganic phosphate group. So to make ATP, we get ADP, adenosine diphosphate, plus energy is converted into ATP, adenosine triphosphate. Let me go back. Any question about this slide here? The generation of ATP is important because ATP is the cellular currency, the currency of cells. Whenever the cell needs energy, it tends to break this bond right here. And if the cell needs more energy, it can also break the second phosphate group. The squiggly line represents a high energy bond, and the third phosphate is bound to adenosine without a high energy bond. Cells have three mechanisms for generating ATP. The first is substrate level phosphorylation, where you transfer a high energy phosphate from the phosphorylated compound directly to ADP. So the phosphate comes off of this molecule and then binds to ADP, and the cell will need energy to make the ATP, but converts the molecule into CCC and ATP. Any questions about any of this? All right, so that was the first way we have for generating ATP, substrate level phosphorylation. Cells can also make ATP by respiration, sometimes called oxidative phosphorylation, but I won't use this term. Cells can make ATP by the sequential transfer of electrons from the electron donor, which in our cells would be glucose, to the final electron acceptor, which would be oxygen in our cells. If the energy were to leave the glucose and go directly into the last molecule in the chain, and the cell at each step along the way can harvest some of that energy and use that energy to make ATP. For our cells, the final electron acceptor is oxygen, which will take the electrons and the hydrogens that are flowing with it and then convert oxygen into water. Along the way, energy is released, and some of that energy can be used to generate ATP. And there's a special process for doing that. It's chemoosmosis. And we'll talk about that probably on Thursday. The third way that cells can generate ATP is by photophosphorylation. Photophosphorylation is only found in photosynthetic organisms. What happens is light releases electrons from chlorophyll, and then the light energy is converted to chemical energy via an electron transport chain, and we'll talk a little bit more about that. In green plants, oftentimes the ATP and the NADH that are generated are used, not always, but are often used to make sugar. And then the store will store up sugar in the cell. Any question about any of that? And here we're seeing two photosynthetic organisms, because only photosynthetic organisms would be covered in mud like that. So the energy that's released from molecules is controlled and stored by a series of reactions. The energy is not released in a single energy burst. And if the energy was released in a single energy burst, the energy being released from a molecule of glucose would be too hot for the cell to survive. And so the cell releases the energy slowly in a series of steps. It's not even showing the first, because the electrons come initially from glucose, which then flows to NADH. Electrons from the donor molecules are passed from one compound to another through a series of redox reactions. For us, our donor molecule is glucose, or some molecule of our food similar to glucose. Most organisms can use as their primary source for cellular energy glucose. But remember, we're talking about carbohydrate catabolism. The cell does not have to make ATP from carbohydrate catabolism. For example, a photosynthetic plant cell would get its energy needs from light and, its energy needs from light. But in animal cells and most bacteria, the most common energy source is glucose. But the cell may use lipids and proteins. To produce enough energy from glucose, microorganisms use two general processes to generate ATP. There's respiration, and there's two types of respiration. Aerobic respiration, where oxygen is used, and then anaerobic respiration, where oxygen is not used by the cell to make ATP. Any question about any of that? All right, and the other way that cells can obtain their energy needs to make ATP from carbohydrate catabolism is by fermentation. This slide is showing you the overview of aerobic respiration on the left, and then fermentation on the right. Let me blow this up. So both fermentation and aerobic respiration start with a molecule like glucose, which enters glycolysis. And then aerobic respiration follows a different path than fermentation. Fermentation takes the end product of glycolysis and then ferments that into another molecule. For example, alcoholic fermentation would convert the pyruvic acid into, sorry, I'm drawing a blank here, into ethanol. The bacteria that make yeast would convert pyruvic acid into lactose, and it will do so by fermenting the, well, mostly the milk, but the pyruvic acid into the fermentation end products. Any question about fermentation? If not, let's look at respiration in a little more detail. It has 3 steps: glycolysis, the Krebs cycle, or I should say the preparatory step in the Krebs cycle, and then lastly the electron transport chain, and it's also called chemiosmosis, but I will use electron transport chain. Essentially, the electrons flow from glucose, a high energy rich substance, to CO2 and water, and these two are low energy rich molecules, and then the excess energy that's given off can be used to generate ATP. How efficient is the cell at forming ATP from one molecule of glucose? The cell is about 40% efficient. And you might say, well, 40% doesn't sound very good. But the best engines we have made can only convert about 20% of the energy in gasoline to the energy of making the car move. So our best engines are about 50% the efficiency of aerobic respiration. Any question about any of that? If not, I think we will stop. Well, let me go ahead and talk about the overview of aerobic respiration. Aerobic respiration is glycolysis, which is followed by the preparatory step and the Krebs cycle. Here in this slide, the Krebs cycle is called the citric acid cycle. And you probably should know both names. It's called the Krebs cycle because it was discovered by Dr. Krebs. And it's called the citric acid cycle because the first molecule made in the Krebs cycle is citric acid. From glycolysis, we do make some ATP, we do make some NADH. From the citric acid cycle or the Krebs cycle, we also make some ATP, and we make a lot more NADH and FADH2. NADH and FADH2 carry the electrons from the Krebs cycle and glycolysis and take the electrons to the electron transport chain. In the electron transport chain, the production of ATP kicks into high gear, and that's when we make the majority of the ATP in our cells. This slide is trying to show you that in a eukaryotic cell, the pink would be all of the cell, and in the cytoplasm of the cell is where glycolysis happens, But the Krebs cycle and the electron transport chain occur in the mitochondria of a eukaryotic cell. Any questions? If not, let me mention that aerobic respiration is also called cellular respiration, and I don't know why there are two names for the same thing, but you really do need to know both names. Cellular respiration means the same thing as aerobic respiration. All right, if there's no questions, I'm going to stop here, and I'll see you at 6:30 for the lab.
Thank you.
All right, goodbye.